Welcome to This Explainer. Today, we're diving into something really incredible. We are going to unpack one of nature's greatest, yet honestly most overlooked structural engineering feats, the hidden, highly complex world of plant tissues. Seriously, if you've ever just stood and stared up at a towering redwood tree, or even just noticed a blade of grass bouncing back after you step on it, you are witnessing an incredibly sophisticated biological machine at work. It's truly fascinating. So today we're going to look under the hood of that biological machine and discover exactly how plants conquer gravity, survive the elements, and even defy biological aging. This is a big topic, and an absolutely crucial one for understanding how life on Earth actually sustains itself. Here is our roadmap for the explainer today. We'll be hitting 1. Nature's engineering marvels. 2. Plants versus animals. 3. The engines of growth. 4. Support and protection tissues. 5. Complex transport systems, and finally, 6, the miracle of totipotency. All right, let's jump into Section 1, Nature's Engineering Marvels, the Hierarchy of Life. Okay, let's dive into this by looking at how life scales up. We really have to ask this fundamental question right out of the gate. What makes cells abandon their solitude to group together and divide the labor? Think about a single-celled organism for a second, like an amoeba. That one cell carries the heavy burden of doing absolutely everything itself. It has to hunt, digest, move, and reproduce all inside one tiny, super fragile membrane. It's a tough gig. But when cells of a similar type group together to form a tissue, they divide that labor. This creates a strict hierarchy of organization, right? Tissues form organs, organs form systems, and systems form those towering, resilient, multicellular organisms we see outside our windows every day. It is the ultimate biological team effort. This drastically increases the organism's efficiency and allows it to carry out some incredibly complex life processes. Moving on to Section 2. Plants versus animals, conflicting blueprints. To truly understand plant tissues, we kind of need to compare their structural blueprints against our own, because the evolutionary problems they are trying to solve are completely opposite to ours. Unlike animals that need cellular flexibility to move, hunt, and seek shelter, plants are entirely stationary. They literally have to face the elements head-on. Because they lack a rigid cell wall, our animal cells can change shape easily, and that's what allows for complex muscle movement and locomotion. Plants, on the other hand, are anchored in place. They require intense, inflexible structural support just to stay firm and upright against crushing gravity and violent winds. Plus, while animals have tissues dedicated to digesting all sorts of different food sources, plants require dedicated tissues to capture solar energy and actually manufacture their own food through photosynthesis. So two vastly different survival strategies naturally require two entirely different sets of biological building materials. Which brings us to Section 3, the engines of growth, meristematic tissues. So how do plants actually build this rigid, towering architecture from the ground up? How does a tiny little seedling become a massive oak tree? Well, they rely on meristematic tissues. Think of these as nature's hyperactive construction crews. These tissues are made up of tightly packed cells with exceptionally dense cytoplasm and thin cell walls. And here is a really crucial detail. They generally lack vacuoles. Why? Because vacuoles are essentially cellular storage lockers, and these particular cells aren't storing anything. Not a thing. Their sole biological purpose is to divide continuously and rapidly to build the plant's body. They are the undisputed engines of pure growth. And this brings us to the apical meristem. These are located right at the growing tips of roots and shoots, and they're responsible for driving the plant's vertical growth in length. Consider this classic biology experiment, placing onion bulbs in glass jars filled with water. Over just a few days, you can physically track the rapid downward growth of the roots. Now, if you were to take scissors and snip off the very tip of those roots, the growth in length would completely and permanently halt. You've essentially fired the apex construction crew, and the plant totally loses its ability to push further into the soil. But obviously, a towering plant needs a massive, strong base to hold it up. And that is exactly where the lateral meristem comes in. While the apical meristem pushes up and down, the lateral meristem adds concentric layers of cells that increase the stem's diameter or its girth over time. If you've ever looked at a cut tree trunk and seen those beautiful annual growth rings, you are looking at the historical handiwork of the lateral meristem. By studying these rings, we aren't just counting the years, we're actually reading a historical ledger. It allows us to decode the exact climatic conditions, severe droughts, and abundant rainfalls of the specific eras that tree lived through. It's like a built-in time capsule. Okay, so we've talked about growing taller and we've talked about growing wider. But what about growing back? That is the domain of the intercalary meristem. These are super specialized tissues located right at the base of the internodes or the stem nodes. And this perfectly explains a phenomenon you probably interact with all the time, especially in the summer. Thanks to these intercalary meristems, grasses can rapidly regenerate and regrow. When the apical tip of a blade of grass is chopped off by a lawnmower or, chewed off by a grazing animal, the plant doesn't just die. Instead, those intercalary meristems kick into high gear, aggressively pushing new growth up from the nodes below the cut. It is an absolutely brilliant evolutionary defense mechanism designed specifically to survive constant pruning and being eaten alive by herbivores. All right, Section 4, Support and Protection Tissues, the Permanent Structures. So, once those meristematic construction crews divide and create new cells, those new cells undergo a critical process called differentiation. Basically, they lose their ability to divide and they transform into permanent, specialized architectural structures. First up, the plant establishes its outer facade, the epidermis. This is a protective, tightly packed single layer of flat cells that wraps around the entire organism. To prevent fatal dehydration, these cells secrete a waxy layer called the cuticle to lock in moisture, which, by the way, is exactly why desert plants have incredibly thick cuticles. But the plant can't be completely hermetically sealed, right? This epidermis houses tiny pores called stomata. These act like regulated breathing windows. They allow gases to exchange and water vapor to transpire, which creates the critical vacuum pull needed to bring water all the way up from the deep roots. Behind that dermal facade, the plant utilizes three distinct, simple, permanent tissues as its core building materials. First, you've got parenchyma. These are the living, thin-walled storage units. They pack away food and house the massive photosynthesis factories up in the green leaves. Second is cholenchyma. These living cells have unevenly thickened corners made of pectin, and they act almost like biological rubber. This provides flexible support, allowing stems and vines to bend in violent winds without just snapping in half. And finally, sclerenchyma. These are cells that have actually died at maturity, leaving behind incredibly thick walls, heavily reinforced with a tough polymer called lignin. This provides massive, rigid structural strength. If you want to know what sclerenchyma feels like, literally just knock on some wood or touch the hard, fibrous outer husk of a coconut. That's it right there. Moving right along to Section 5, Complex Transport Systems, Nature's Plumbing. Now, a massive redwood tree can't survive just on solid structure alone. It needs an intricate plumbing and logistics network to transport resources up and down its towering height. This visually striking network is the xylem. It's a powerful upward pumping system made mostly of thick-walled tubular cells that, again, are entirely dead at maturity. Because they're dead and hollowed out, they form perfect, unobstructed microscopic pipes. They transport water and dissolved minerals from deep underground, fighting gravity for hundreds of feet into the air, utilizing that transpiration pull from the leaves we talked about earlier. It is a jaw-dropping engineering marvel of fluid dynamics. And running strictly parallel to the xylem is the phloem. While the xylem pumps water up through those dead pipes, the phloem is a living logistical network. It's made of sieve tubes, which are tubular cells with perforated ends that expertly distribute the sugars manufactured by photosynthesis in the leaves all the way down to the roots and the rest of the plant. Now, because these sieve tube cells are so highly specialized for transport, they actually lack a nucleus. So their life functions are actively monitored and kept alive by adjacent companion cells. These companion cells basically act as the biological dispatchers, regulating the flow and ensuring this living highway runs smoothly at all times. And that brings us to our final part today, Section 6, the miracle of totipotency, cellular regeneration. So the truly crucial point here is that while tissues like the xylem, phloem, and epidermis are highly specialized and permanently structured to do one specific job, nature has one final, astonishing trick up its sleeve. Enter totipotency. This is, without exaggeration, one of the most mind-blowing concepts in all of biology. Totipotency is the incredible ability of a mature, completely specialized plant cell to just hit the reset button. It can reverse its programming, lose its specialization, start dividing like a meristematic cell all over again, and literally grow an entirely new, whole organism from absolute scratch. This isn't just theory either. A scientist named F.C. Stewart famously proved this using a simple carrot. First, he extracted highly specialized mature phloem tissues from a carrot root. Second, he cultured those tissue fragments in a nutrient-rich liquid medium.
In that environment, step 3 happened. Single cells sheared off, completely undifferentiated, and started dividing rapidly. This led right into step 4, where those cells redifferentiated, forming roots, shoots, and eventually an embryonic plant. Finally, from that single specialized cell, stewards successfully planted and grew a perfect, fully adult carrot.
Which leaves us with a pretty staggering possibility to consider as we wrap up this explainer. If a single, microscopic, specialized cell securely holds the entire architectural blueprint to rebuild the host organism from absolutely nothing, the potential here is limitless. How could our ongoing efforts to master this hidden architectural flexibility completely redefine the future of global agriculture? I mean, how could it secure crop improvement in a rapidly changing, unpredictable climate? And ultimately, how could it safeguard human survival in the decades to come? It's a really profound thought to take away. Thank you so much for joining me on this deep dive into the hidden and incredibly important world of plant tissues. Keep questioning and keep exploring.